Good evening. We are so happy that we're together today for the PSC Partners Roadmap 301 research meeting. The PSC research landscape now and into the future, charting a path forward. My name is Joanne Hatchett, and I'm Medical Science Liaison at PSC Partners. It is so exciting that so many of you have joined us this October evening. This meeting will start with a moderated panel discussion about the future of PSC, followed by moderated breakout rooms for Q&A and discussion. Next slide. Before we get started with this Roadmap 301 meeting, I'd like to briefly review what the Roadmap Initiative is all about. Roadmap is one outcome of work that PSC Partners has been doing over the past few years. This includes the receipt of a Rare as One grant. A primary goal of this grant is to leverage the power of patients to accelerate PSC research, including the development of a patient-organized collaborative research network. In addition, last October in 2020, we had a PFDD meeting on PSC with the FDA. And we have our PSC Partners Patient Registry, which currently has over 2,000 participants from around the world. Together, this work has led to expanding the vision of the role of PSC Partners and the PSC patient community in research and drug development. Next slide. With that bit of context, let's revisit the goals of Roadmap. The goals are to accelerate progress, finding therapies and a cure by educating our patient community on the research landscape, broadening the researcher base, and strengthening multi-stakeholder communication, creating opportunities for researchers and clinicians to engage in meaningful discussions with an informed patient community. Next slide. Practically, what does this mean? Hopefully you've seen this roadmap graphic, which to the right shows our three main stakeholders. On the left, you'll see 4 topic areas as steps. Currently, we are on topic 2, research. For each of these topics, we are hosting a series of meetings with 2 or more Zoom meetings for each topic. The one-on-one sessions are educational and community focused for PSC patients and caregivers, and the 201 and 301 sessions are multi-stakeholder discussions with PSC researchers, clinicians, and pharma. Next slide. Thank you for joining us. And again, welcome. You'll see our agenda here. And now I have the pleasure of passing the Zoom mic to Ricky Safer, founder and CEO of PSC Partners Seeking a Cure.
Hello. Thank you, Joanne. And thank you to all of tonight's attendees, both first-timers and repeat attendees. This is the 3rd and final research webinar in a roadmap series, as Joanne said, before we move on to the topic of drug development. Thank you to our sponsors, Albireo, Intercept, Escient, and Mirum, for your support of our ongoing roadmap initiative. Here's today's agenda. Our 4 expert panelists, led by Dr. Lisa Forman, will review what is known about the role of bile acids, genetics, microbiota, and immune cells, and the interplay of these concepts in PSC research. They will also give us their opinions on avenues for future research and suggest key unanswered research questions that need to be addressed. This will be a lively panel discussion. Now I'm pleased to introduce tonight's expert panelists and moderator. Dr. David Assis is an assistant professor of medicine, section of digestive diseases, medicine department at Yale University School of Medicine. His clinical and research interests are in autoimmune liver diseases, specifically focused on autoimmune hepatitis and primary sclerosing cholangitis through both clinical work and translational and basic research. Dr. Assis is also a member of our Scientific Medical Advisory Committee. Dr. Angela Cheung is a clinical hepatologist at the Ottawa Hospital and an assistant professor at the University of Ottawa's Faculty of Medicine. Her work uses artificial intelligence to fight liver cancer by training computers to recognize it using images from patients with or without liver cancer. Dr. Richard Green is a professor of medicine and hepatologist at Northwestern University Feinberg School of Medicine with a clinical interest in the medical care of patients with PSC and other cholestatic liver diseases. His research focuses on the molecular mechanisms that cause liver disease and applying this knowledge to develop novel therapies for patients with PSC and other liver disorders. Dr. Green is the co-chair of our Scientific Medical Advisory Committee. Dr. Alex Miethke is a pediatric gastroenterologist and director of the Center for Autoimmune Liver Diseases at Cincinnati Children's Hospital. His NIH-funded research focuses on the control of hepatic immune responses by bile acids and bile acid receptors. He co-chairs the Children Pediatric PSC Observational Study, which is soon to start enrollment. Children stands for Childhood Liver Disease Research Network, which is a multi-center NIH-NIDDK supported consortium of 12 pediatric hospitals across the US and Canada. Tonight's moderator, Dr. Lisa Forman, is professor of medicine in the hepatology section at the University of Colorado. Her main clinical and research interests are in autoimmune liver diseases, including PSC. And now I'll turn it over to Dr. Forman to start tonight's panel discussion. Thank you.
Thank you, Ricky, for inviting me to moderate tonight's session. I think it will be very exciting, and hopefully we can spread a lot of updates and what's current and upcoming in research, as well as get the audience ideas of what they think is most important to them moving forward to help us come up with new ideas and new avenues for research. And so, as Ricky said, there are some basic areas that we hope to cover today during this panel discussion, including what is currently known, new information about PSC, where we can go in the future, as well as why it's so important for you as patients and caregivers to have a voice in this process. And so I'm going to go over the list of questions, and instead of just calling initially on each panel member, hopefully you can each chime in on what you think is important, especially if it's your area of expertise. So with that, I'll begin. And the first question is really, how has research over the past 5 to 10 years shaped our understanding of the mechanisms contributing to the development or progression of PSC? So if one of you could please comment initially specifically about the role of bile and bile acids. I'm going to have to call on people if no one volunteers.
Okay, I'll start. So
thanks, Dr. Green.
It's been known for decades that bile acids are very important in a whole host of liver diseases, certainly in PSC. And there's a healthy bile acid called ursodeoxycholic acid that was used for several decades, and because in general for many cholestatic liver diseases, it makes the bile healthier and less liver injury. Several years ago, it became clear that high dose urso is contraindicated in PSC for reasons, some of which we understand and some of we don't understand. I think the two main issues that have come up that offer promise are, first of all, the microbiome. The germs in our gut actually metabolize bile salts. They change one bile salt into another. And I think it's well accepted that the microbiome, these germs, are very important, although we certainly don't understand all of it. The other thing that has become evident the past 15 years is that bile acids, which were thought for years to be simple detergents, regulate a whole host of genes, which can affect the function of the liver, scarring of the liver. So both these offer new avenues for investigation and potentially therapeutic targets.
Any other comments from the panel?
I would like to add to it. I totally agree with Richard that perhaps especially over the last 3 to 4 years, the perspective on bile acids has also changed a little bit. I think in the past it was studied a lot how bile acids, the retention of bile acids in PSC would cause cell injury, cholangiocyte injury, hepatocyte injury, various injury mechanisms to the liver itself. As Richard pointed out, it becomes now much clearer that the bile acids have a very important regulatory function. And over the last 2 years, there were 2 very exciting papers from high-profile investigators, Littman's group and Rudensky's group, showing that bile acid metabolites, as Richard pointed out, the bile acid being metabolized by the microbiome in the gut, that these bile acid metabolites very closely, tightly control the balance of regulatory T cells and Th17 cells in the gut. And they may have an impact on a host of autoimmune disorders, including PSC, although that has not really been studied, how the effect of bile acid metabolites, and subsequently the immune responsive originating from the gut are directly related to PSC. But I would foresee a lot of interest in better understanding that connection, then potentially targeted with therapeutics, because in these studies by Littman and Rudensky, they would feed the mice with the bile acids which had a beneficial effect on the Treg balance, or they would give bioengineered microbial communities to enrich the intestinal bile acid pool size with advantageous bile acids to promote induced regulatory T cell responses. So I think the field in the basic science is already really advanced. It remains to be seen how we can harness those findings for clinical trials.
Yeah. So have those studies kind of for the group been replicated at all in humans?
You know, that is, in my mind, a little bit the problem when you ask a 10-year perspective. I think we have a lot of clues going in certain direction, bile acids, immune, certain bacteria. But when it's about replicating, then we still face challenges. You know, when I think the PSC partners sponsored the fetal matter transfer clinical trial from the Boston group, right? And they had some positive findings, but they couldn't really show that anything changes with the bile acid, with enriching healthy bacteria, didn't really change anything on the bile acid. When the recent gastroenterology paper by Crumman on the metagenomics looked at the metabolic genes, there was nothing about bile acids. There was about vitamin B6 and branched amino acids. Bile acids, there was nothing. So I think we go in waves and we will come back and we will converge to the truth. But I in my mind, there are still a lot of loose ends. Nice, interesting targets, but I don't have quite the smoking gun here. Maybe David has it.
I'd love to have a smoking gun, but yes, I will add to what Alex and Richard said, which is exactly right, that we are still being surprised, I think, constantly with some innovations, and sometimes clinical advances also help to confirm some pathophysiologic scientific truths as well. I think all of us would agree that We don't yet have an effective therapy that we wish to see for PSC, but there are several compounds, medications, drugs that are moving forward in clinical trials. One that is not yet available and not yet approved, of course, but it's being studied in Europe would be norursodeoxycholic acid. And norurso would be one potentially interesting molecule. It potentially has some advances over the more traditional ursodeoxycholic that Dr. Green had talked about in terms of a beneficial for as a choloretic agent as an anti-inflammatory. And I'm particularly intrigued by some recent studies by the group Michael Trauner that norursodeoxycholic acid seems to have a beneficial effect on T cells, which we know are very important in PSC. And so I think some promising molecules may allow us to potentially have some benefits for the bile acid pool, which we know it needs to be fixed in PSC, so to speak, but also on the immune cells. So sometimes when a molecule moves forward clinically, we also get a chance to study how it works on more than one mechanism. So these kinds of stories, hopefully this will be a successful story, I think teach us a lot also on the scientific side.
And, because PSC pathogenesis is so complicated, obviously there are many different factors that are contributing, which is also why I think drug development has been a little bit more challenging. If someone could maybe comment now specifically on something that has already been commented on, the microbiota, maybe as it relates to patients with PSC alone versus PSC in patients that have inflammatory bowel disease. Is there any difference in these studies when you look at someone who's an adult with PSC versus a child?
So I think what's been interesting in the microbiota studies is that there haven't really been significant differences between patients with PSC and IBD. Some of that might be driven by the fact that patients with PSC might have sort of mild IBD that's been undiagnosed. I think it's interesting, though, it really speaks to potentially the close relationship between PSC and IBD, because we know such a high prevalence of PSC patients have IBD as well. But I think also it sort of speaks to what Alex, I think, was alluding to a little bit in that, we haven't yet found the smoking gun, and Lisa, you alluded to this as well, is that there's so many factors that come together likely to cause PSC that I don't know that we'll ever find one smoking gun. I suspect there's multiple phenotypes of PSC. We all know, I think, that there's some people who progress slowly, more slowly than others. And I think it'll be interesting to see what distinguishes those people in terms of microbiota, bile acids, immunity, even metabolism. And I suspect we may find signatures that may combine a variety of these things that sort of drive people in terms of their progression or a lack thereof. And so I think it is going to be a pretty complex answer. And while I'd also like Dave, and I think everyone else here would love to find a smoking gun, I think the human body is so complex that it's going to be a combination of things. And if we're able to find sort of profiles or signatures that speak to that, hope that we'll be able to find multiple medications, for example, sort of like how we treat hepatitis C to do sort of multimodal or a multi-drug approach.
Have there been any studies looking at FMT in patients with PSC?
Small study, go ahead, Alex.
David, all yours. You're close to Boston.
Good point. So a small study by the group for which Josh Korsnick is involved, but Jessica Allegretti, a small study of about 10 patients or more from last, I think, 2 years, had studied this in the context of PSC. I completely agree with Angela that smoking gun may be premature to search for in this area, but there was differences in the composition of the microbiota before versus after, maybe some small trends toward differences in liver enzymes, but I think it was, in my perspective, more of an exploratory study about the feasibility, perhaps the reaction, and I think oftentimes safety, as we all know now with fecal microbiota transplant, is very important. There was no concerns on that end, but the long-term effect of that on clinically meaningful parameters that we know to be important in PSC remains to be determined.
And can someone comment on the role of genetics?
It's clear that PSC has a very strong genetic component. And, it's not a classic genetic disease. And in fact, most human diseases are not due to one or two genes changing like we thought perhaps that was the case several decades ago. It's due to a whole host, we call them polygenic diseases, meaning several different genes interact with the environment. And there have been some large, what they're called GWAS studies, which are studies that allow us to look at large populations of patients with a disease such as PSC and identify some of the contributory to genes. Again, there's no smoking gun, there's no PSC gene. It's a combination of many genes along with the environment and all these other factors.
And anything specific anyone would like to discuss about the immune system?
I mean, I'm always intrigued by the fact that, as with other autoimmune diseases, unfortunately, PSC can recur in after transplant. As much as we're unfortunately used to thinking about that and seeing that, there is no doubt that if the bile ducts can become damaged, and someone else's bile ducts can become damaged after brand new liver is put in, there is a very strong immune loss of tolerance that those immune cells, again, partially genetically driven, probably also environmentally, degree of colitis and other things, but there are immune factors that can attack even a brand new liver or a second liver and a third liver. And so I think that one of the frustrating parts from my standpoint is the lack of effective immune modulating agents for a condition that is very clearly driven by the immune system at key points in the pathophysiology.
Agree. Now, I know Alex had mentioned some literature that he found intriguing. Are there any other recent publications that you could share with the group on this topic? And then maybe Ricky can kind of post the articles in case any of the researchers and patients that are in our audience can look up if they'd like to.
I think one paper, although it's already 2 years old, but I think it's probably well known to many here. But I still think it's one of the most elegant works I have seen on that topic is from the Nakamoto et al. in Japan, and it basically summarizes a little bit what we have been talking about. And that group isolated the intestinal microbiome from several patients with PSC, and through a series of experiments, they ended up injecting germ-free mice with the intestinal microbiota, and they could see that they would induce a phenotype similar to PSC. There are a lot of other things they did, but in the end, they boiled it down to 3 species: Klebsiella pneumoniae, Enterococcus, and Proteus. And when they used only these 3 bacteria, they could also induce a phenotype of PSC when it was combined with a toxic bile injury from DDC. And they showed that they 3 bacteria had to work in concert to do that. The Klebsiella would sort of cause pores into the intestinal layer and allow translocation of the other bacteria into the mesenteric lymph nodes. Then there would be a Th17 response which would be generated, and then that would lead to ultimately the liver disease. And at each site, you could interfere and block the progression. You can treat them with oral vancomycin, and you would kill off the Enterococcus, and you wouldn't have the result. You can block the Th17 pathway with an anti-IR gamma T antibody, small molecule, and you would block the progression. So it very nicely showed how that infection, which many of us believe is at the basis, otherwise it wouldn't recur after transplant, is linked to an immune response. And at least two of the topics you ask us to comment on, they were involved there. But now we come to the problem. Klebsiella pneumoniae, at least that one, that is not a very abundant taxa that were discovered in these microbiome studies. So there are already bacteriophages available, viruses which can kill this type of bacterium, and it has been entertained as a clinical treatment in IBD. Maybe we just have to do some of these trials and learn from the trials if they are safe, and have to study very carefully what these interventions do to really figure out what is causal for PSC, which will be the next step from all these observational studies we have done over the last 30 years.
So I think so far, everyone's done a really nice job in kind of explaining and shedding light on why PSC is such a complicated disease, why it's been so complicated to study, and why it's been so difficult to come up with, as you all put it, the smoking gun, or one drug that fits all. Moving on to kind of the future, what area of PSC research excites you the most? David, I'm putting you on the spot.
Thank you.
You're welcome.
I have to say, I do think that the interplay between pathogenic bacteria and the gut and their ability to translocate to the liver or to generate a pathogenic immune response is the most exciting area for me. I share Alex's enthusiasm for the Nakamoto paper, and our group is starting some studies of bacteriophages also to target some of these bacteria. I think it's very exciting, and it also opens a lot of new cans of worm, to be quite honest. The one, some of the studies, although they're in mice, of the MDR2 knockout mouse, if you take away all the bacteria in that particular model, the mice do worse with regard to bile duct injury. So clearly it's not so simplistic as to just wipe out all of the bacteria. But I truly think that the interplay between the environment represented by the bacteria, the immune system, and the effect on the liver is the most exciting area for me at the moment, beyond the perhaps promise of genetics from 15, 20 years ago that I don't think we've yet found nearly enough of an answer, therapeutically at least.
And Angela, do you think that there is a particular area right now that is underappreciated and maybe should be prioritized for future research in PSC?
Yeah, I think it plays into a little bit about the paper that Alex had mentioned earlier, which is the group from Norway and Germany, Dr. Carlson and Dr. Schramm's group, where they looked at sort of the metagenomics of the genes of the bacteria, and then also their function along with the metabolites. So I think metabolites is sort of one feature that is sort of maybe missing from the list currently, and I think might prove to really provide a lot of strong evidence on pathogenesis of PSC. So if we could include that along with microbiome research and bile acids, because I think that study also highlighted the importance of doing sort of concomitantly all of these sort of samples. So they hadn't taken blood at the same time as stool. And so it was a bit difficult to make a direct comparison between what was occurring in blood and how that might be affected by the microbiome. And I think, Alex and David both mentioned it, sort of to try to come up with an answer and understand how these factors are affecting each other. We really do need to have concomitant simultaneous sampling. And then there's also like analysis of the samples in terms of the microbiome, metabolome, et cetera. And I think one other interesting area is the immunometabolism, where we know now that there's actual like significant metabolic pathways within immune cells, and alteration of these metabolic pathways also really alter immune function. So I feel like metabolites is sort of a huge area, which we could definitely use more focus in, because it affects both potentially microbiome and then may connect to the immune system as well.
And then Richard, maybe you can just kind of summarize what you think are the kind of key unanswered questions right now that need to be addressed, that you think will have the most impact on patients with PSC.
Again, I think the microbiome is we're in our infancy of understanding it. It's intuitively obvious why the microbiome, the guts, flora, bacteria would affect the liver and the gut. But in fact, the microbiome affects the heart, obesity, lung disease, virtually every organ. So there are many systemic effects of the microbiome that we're just now beginning to understand. Along what Angela said is metabolomics, which are kind of the small molecules that these bacteria make. It was very difficult up until fairly recently to and very expensive to analyze the chemicals these bacteria were making. And now with mass spectrometry and other techniques which have become much cheaper and much more available, I think that's really an avenue to investigate in a way that we couldn't do it effectively or cost effectively even a few years ago.
And moving just to the last section of the panel discussion, I'm sure we'll get more into this in the breakout rooms. But I think we all know that the PSC population in general have been wonderful advocates for themselves, especially groups like Ricky's, and have really kind of led to a lot of these research. So focusing now kind of on the patient voice, if the panel can maybe comment about how we can best communicate the importance of patient needs to the rest of the medical community. You know, how can we ensure that the patient voice is heard and incorporated in the future for research? Open this to everyone.
I think part of it is to have very thoughtful patient advocacy along the way to make it clear what are the parameters that make for a successful study. I think among many other things, studies that are feasible and that do not place undue burdens on the patient and that can really incorporate quality of life, ideally into some of the outcomes, I think are a win-win, because we focus on the clinical endpoints, the 10, 20-year outcomes. But if somebody feels miserable on a therapy for that entire time, that may not be a great trade-off. So I think helping the design of studies that are patient-centric, but also incorporate the patient voice is one way that I think we can work together.
And I know we've had addressed this in some of the previous research seminars on patient-reported outcomes and really incorporating those in all these other research that we're doing. Of course, a lot of what we talked about today is not even been applied yet in clinical trials. It's, again, at the basic science level. But as we move forward, incorporating what's important to the patient in terms of their outcomes, their quality of life is going to be just as important. Any other comments from the panel about how we may better work together with the PSC community to address their needs?
Maybe I can share from the pediatric perspective, because we are really blessed. We not only have the patient's voice, we also have the parent's voice. And that is probably amplifying signals. At least that's what we experience. So we learned this, and we have had patient sort of directed symposia in Cincinnati, and now we founded a learning health network. And as one example, we do hear loud and clear the parents' stories about successful oral vancomycin treatment, despite negative retrospective studies, which David also commented on in editorials. And I think that is important. I think it helps us to push this forward. We cited a few basic science papers which would suggest that there is a rationale for certain antibiotics to work. And it may not be an oral vancomycin in the future, but it is probably a targeted therapy for bacterial communities or metabolic byproducts of these bacteria. But I think it is great to have narratives and success stories pushing forward the field. And I think the pediatric hepatology community is very receptive to these voices.
The other thing that I think we look to the PSC community for is prioritization of what they value. If all resources are put to where we can cure PSC in 30 years, that's great, but it's not going to help people now. So it's a trade-off between quality of life and therapies now with longer term studies that may give us answers in the future. And it's really the PSC community, people living through this or have loved ones who are living through this, who we look to prioritize how we should be studying this.
So thank you so much to our moderator and panelists tonight. This has been such a fascinating conversation, and we so appreciate you all sharing your time with us.
Okay. I'd like to thank tonight's panelists and co-facilitators for making tonight's webinar such a success. I know our breakout group was great today. And thank you to all the PSC patients and caregivers, medical professionals, and industry representatives who participate in the breakout room discussions. In advance of our June 2022 conference, where we will launch our International Collaborative Research Network, these webinars are a venue to open up meaningful discussions between the various stakeholders. I hope you all enjoyed today's discussions. Here are a few reminders about our current and future programs of interest. Visit our website at pscpartners.org. Participate in Global PSC Awareness Day this Friday, October 29th. We're really excited because for the first time, this will be an international event with 12 PSC patient organizations working together. Watch for social media for ways to join in the fun. Please save the dates June 2nd through 5th, 2022, and plan to attend our conference in Boston. We are planning this as an in-person meeting after 2.5 years of being virtual, combining our traditional PSC patient caregiver conference with the addition of an International Collaborative Research Network component. You won't want to miss this. Join the patient registry if you haven't already. Ideally, we would love to have every PSC patient participate to help us accelerate PSC research. Listen to our monthly podcast created by award-winning filmmaker Niall Miethke. Visit links to our updated COVID-19 and vaccine information. We have Zoom rooms coming up. So the first one is adult PSC patient ages, 18 and up, will be on Tuesday, the 10th of November. Caregivers, spouses, and parents will be Wednesday, the 1st of December. And teens 13 through 20 will be the 14th of December. So we hope you will join these. Canadians also visit pscpartners.canada and sign up for their mailing list and follow them on social media. And I'm going to turn it over to Ruth Ann to tell you a little bit more about that and an update. Okay, Ruth Ann.
Thanks, Ricky. So before we leave tonight, I want to briefly summarize the Roadmap Research Series. We have accomplished so much together through this Research 101, 201, and 301. Over these 3 webinars, we've welcomed more than 250 participants. Thank you so much for being a part of this mighty community. You've all voiced dozens of research questions, and we've been collecting all of them. We've also progressed our understanding of the PSC research landscape, from understanding the basic PSC topics to developing our scientific skills and engaging together to identify key unanswered questions and unmet needs in PSC. But this is really just the beginning. We'll prioritize PSC patient needs in developing a strategic research plan and launch the PSC Partners International Collaborative Research Network at the 2022 conference. We hope to see you all at the Roadmap Drug Development 101 on December 8th. Have a great night, everybody.